Hello everyone. So for the recap about our topic, which is gene therapy. So what is gene therapy? Gene therapy is a treatment or cure for disorders caused by mutated genes. It involves adding a normally functioning copy of a gene to affected cells to restore the normal function. So this means that instead of using regular medicine, so gene therapy focuses on fixing the problem at the genetic level. So when a gene becomes damaged or mutated, so the body cannot produce the correct proteins that needed for the normal function. So by inserting a healthy gene therapy helps the body work properly again. So gene therapy is different from traditional treatment because it targets the root cause of the disease, not just the symptoms. So who is the father of gene therapy? So the father of gene therapy is William French Anderson. He earned this title because he led the first gene therapy treatment in 1990, where a girl with a rare genetic disorder received a healthy gene to improve her condition. So, is gene therapy safe? So, scientists are still studying gene therapy to determine how safe and effective it is. Many patients have shown improvements, but there are still risks such as the immune reaction, the inflammation, and or the unintended changes in DNA. However, modern techniques and are carefully monitor the clinical trials and helping to make gene therapy safer over time. So the process of gene therapy involves using the vector. So usually a modified virus to deliver a new gene into a patient cells. So first is the new gene. So the new gene is the therapeutic gene is prepared to be delivered and inserted into the vector. Then second is the vector. It is a harmless virus used as a delivery vehicle to carry the new gene. So it attaches the target cell and then ato na po siya sa vector binds. So anina, which is the virus sticks to the outside of the cell. Then after that, it will go to the package vesicle, which is the cell pulls the vector inside, then through a small bubble that is called a vesicle. Then next is the vesicle bricks, which arena ang bubble ma-open, then releasing the vector into the cytoplasm. Then lastly is the vector delivers the new gene into the nucleus, where it begins the function and help to restore the normal activity. So, or normal cell. So in most gene therapy procedures, viruses are used as carriers because they naturally known as the human cells. So these viruses are genetically modified, so they cannot cause diseases and will only carry the therapeutic gene. So there are two types of gene therapy. First is the somatic gene therapy. So it is the transfer of DNA to any cell of the body that does not produce sperm or eggs. So it will affect only the treated person. It will not be passed to the patient's children, and it is the most widely used. Kaduha, nga types is germline therapy. So it involves transferring DNA into the cells that produce eggs or sperms. So it affects the effects can be passed on the patient's future. the future children. So kung ang changes nga mamugna in the reproductive cells, so it will be inherited by the next generations. But it is controversial and restricted due to the ethical and safety concerns. So in summary, gene therapy is a way to treat genetic diseases, so by fixing the genes themselves, so not just the symptoms. So while it shows great promises and ongoing research, it is essential to ensure it's safe and effective for patients. Thank you.